big guns, upstream analysis. These are our godfathers. 15 minutes left. Okay. Here is this table. These are all our upstream regulators. This next column is if we actually measured if they were present in our gene list. If you look, most of these top regulators that we're finding were not. And that's pretty much standard. Like I said, if I want to get somewhere fast, I don't have time. I'm not going to build a car. I'm going to take a cab. And that's exactly what your cells do. That these things are more active, and you're not seeing it at the transcript level. But again, these things are making waves in our transcriptome. So this top one, SOX2, and you can Google that, SOX2 and stem cells. You're going to see tons of stuff. This is a big time player. And in fact, this is probably is more responsible for the embryonic state. What we find is that 38 of our genes are known to be affected by SOX2. And we can actually select that and display as a network. So you can see SOX2 in the middle. And these are all the genes it's known to interact with in our gene list. The odds of getting all of these genes by random chance, by picking out all these pink balls, is like 10 to the negative 13th, I think, if I remember right. These colors mean something. That SOX2 is known to inhibit axin 2. It's saying that this is an activation relationship because this is going up, so therefore SOX2 must be going down. Again, anything in yellow is not following the trend. Ooh, there's DUSP1. Again, as you go through these things, you just find these connections. I just found this now. So we've got a known effector for the embryonic state interacting with a known inhibitor of the EMT transition. Here's the deal is you could always go upstream. You can go farther and farther and farther down the rabbit hole. In fact, there's a Buddhist saying, the farther you enter the truth, the deeper it gets. You can get pretty deep with these transcriptomes, I'm telling you. All right. Okay. This is what kind of molecule it is. You can see a lot of transcription regulation. Again, these are probably the things you want to target, right? You don't want to target the last domino. You want the ones at the beginning. Transcription factors are really good upstream regulators, right? They basically induce the expression of lots and lots of genes. Let's see. So we got that. We have our activation state. We can sort by that, so do that. These are the upstream regulators more active probably in our pluripotent stem cells. And if you look these up, these make sense. Estrogen, that's an interesting one. Ooh, there's the EGR. Remember that? We saw that on the EMT transition. We were seeing that this is probably up because of its signaling molecules, but it's getting bottlenecked somewhere. What's inhibited in the pluripotent stem cells? So these are all the things that are probably more active in our embryonic stem cells. Not only that, but we actually measured a couple of these. Yeah, fold change isn't incredibly great, but you can see that this serum response factor, we measure it going down, and sure enough, the software says, yeah, it's probably going down, based on the expression of the 20 genes that it's known to interact with. This one, I'm not so certain. This is different, right? It's saying it's going up, but it's also saying it's inhibited. There's only 6 things known to interact with it. I would believe these 20 versus this 6. Serum response factor might be a great target. I'm not going to have time to do the whole using that 25 more, but oh well. Okay, sort by p-values again. I want to show you one more thing. Make sure you're at the lowest one. Does everybody have SOX2 as their top upstream regulator on your list? Okay, that's good. So we basically here's the p-value where that's a probability that you might get that by random chance. Again, here are all the molecules on your list that are known to interact with it. And it's basically showing you whether they go up or down. Now here's where you put the godfathers together. This last column, mechanistic network. What it does is instead of grouping the genes based on some, their relationships, what it's doing is it's grouping all these upstream regulators into their own networks. This is where you get to the skeleton of all the gene expression. This is the heart of all the things that make all your stuff change. Click on that. TGF-beta. I see TGF-beta in anything that's having to do with epithelial cells or skin-like cells. TGF-beta is playing a role. I'm going to tell you it's all about cell adhesion. That I analyze data sets. Anybody heard of pulmonary fibrosis? It's a disease Evel Knievel got, or had. Now he's dead, but something you can't cure. It's basically uncontrolled scarring. Cell adhesion goes nuts. These cells attract each other, stick to each other. This thing is going down in our pluripotent stem cells. Not only that, but it's probably affecting all of these molecules as well. SMAD3 is a huge secondary messenger of TGF-beta. If you look up SMAD3 in embryonic stem cells, there's a lot of literature on that. SMAD3 is usually pro-embryonic. We're seeing that SMAD3 is probably down in our pluripotent stem cells. Again, these are all the rock stars of stem cell research, actually. Beta-catenin, EP300. This affects chromosome condensation. Interferon gamma. This is a total big-time inflammatory response. Angiotensin. So basically, and I've seen that a lot too, these pluripotent stem cells are producing genes to start making blood vessels. We don't want that. Not yet, anyway. Again, this is giving me evidence. This is showing me that, yes, these two cell types are different. Not only are they different, but the pluripotent stem cells, like we would expect, are more differentiated than probably the embryonic. That's the big thing from this. Number two is, I've got some things that I think might be causing that I could potentially manipulate. That if I went to my collaborator and I said, let's create a pluripotent stem cell more like ES cells, I would say, hey, maybe we should upregulate TGF-beta1. Not only that, but there's some other things that I think would be very interesting to play with. Some of those other targets that I got. And you can do that. That's the thing, is we can test all this stuff. If I think something, if I inhibit something and should make it, more embryonic-like, you can get, shRNA against that gene. You can get antibodies that block it. You can get all kinds of stuff. All right. Let me show you one more thing. I got 7 minutes.